Today, I will be breaking down a research article on immunotherapy. It uses both toxin, which is called diphtheria toxin A, and a foreign antigen. So, what's the difference between chemotherapy and radiotherapy from immunotherapy? So chemotherapy and radiotherapy, they use both. They use drugs to attack both normal and cancerous cells. So that's going to cause a lot of damage to the normal cells. And the patients may experience side effects such as hair loss, fatigue, and nausea. Whereas immunotherapy, it uses external stimulation to boost the patient's own immune system. So it has better precision, adaptability, and long-term memory, because we know that immune cells can have memory cells such as memory B cells, memory T cells that can generate secondary immune responses faster and more efficient when the same antigen is presented. So the challenges for current immunotherapies are: first, tumor cells can hide from the immune system. For example, they can evolve to downregulate their expression of the antigen on the surface of their cell. So the chances that they are going to be recognized by dendritic cells is decreased. Secondly, the endogenous tumor antigens are hard to identify and can vary between patients. Lastly, immune suppression in a tumor microenvironment limits effectiveness. For example, tumor cells can excrete cytokines such as interleukin-10, interleukin-8 that can regulate the immune system. For example, tumor cells can let macrophages to be turned into M2 macrophages that would not kill the cells, but it works mainly in repairing damaged tissues and building new blood vessels, which are favorable to the tumor cells. And in this article, they made a hypothesis. What if they could use a light switch transgene system to make cancer cells self-destruct and train the immune system to hunt these cancer cells down? So they use a light switch, a blue light switch, to control the expression of the toxin DTA and introduce foreign antigens to boost the immune system against tumors. Now let's break down the scheme graph. Two mechanisms. First, the blue light switch transgene system that are introduced into the cancer cells. When we switch the blue light, it's going to express DTA, and DTA is going to cause cancer cell apoptosis and also release tumor-specific antigen that lives inside the cancer cell that cannot be detected by dendritic cell and activate T cells. So now it's being released, it can activate T cells and generate immune response. The second mechanism is to use foreign antigen, OVA, to activate T cells. So how do we transport DTA and OVA into the location we want? If we think about that, of course we want DTA and OVA to reach to the tumor site, because that's where DTA can do its job to kill these cells, and OVA can arrive at the surface of the cancer cells to activate immune cells. And also we need a part of OVA to be transported into the lymph node system, because that's where T cells live. So we need the antigen OVA to be grouped with the MHC structure on dendritic cells to activate T cell, and let T cell leaves the lymph node to the tumor site and to kill any cancer cell that has same antigen, which is OVA. So we do this by introducing nanoparticle delivery system. The first one, how the light control transgene system works. So I'm just gonna explain it briefly, but if you want to know more detail, you can search for this article to learn how this whole thing works. But in short, they use a transcription factor called GAVPO. So in the dark, GAVPO is inactive. But when exposed to blue light, it undergoes a conformational change, a dimerize, that allows it to bind to a specific DNA sequence called UAS, the upstream activating sequence. Once GAVPO binds to UAS, it activates downstream promoter, which is the promoter for DTA. And DTA kills the cell by blocking protein synthesis and lead to cell death. So by controlling light exposure, we can selectively trigger cell apoptosis. And just a reminder, all of the GAVPO and DTA DNA are in a synthetic plasmid. So from plasmid to protein, we need to first transcribe the DNA into mRNA. And that's why we need a promoter. When a promoter is activated, the RNA polymerase will move to this place and start transcription. And then the mRNA move to the cytoplasm of cancer cells to be translated into protein, which is DTA itself, to do its job to kill the cancer cells. And this is their design of the nanoparticle delivery system. So they use PI, which is polyethylene amine, and V as vitamin E succinate to form a polymer network. They modify another layer of hyaluronic acid, PG, polyethylene glycol, and RGD, which is free amino acid. And the hyaluronic acid here is because hyaluronic acid can bind to the receptor on a cancer cell surface called CD44, and they can let the nanoparticle delivery system to enter the cancer cell. The whole process in general is first we fabricate the nanoparticle delivery system, and we inject this nanoparticle delivery system into the tumor cell. And then we turn on a blue light and a DTA be expressed and generate DTA-mediated cell killing. And in this process, the tumor-specific antigens will be released and lead to cancer cell apoptosis and also generate an immune response. So how did DTA enter cancer cell? It's not like by injection only it can enter the cancer cell. So it achieved this by receptor-mediated endocytosis. The nanoparticle delivery system, we already mentioned that hyaluronic acid will bind to the CD44 receptor and enter into the cancer cell because the cancer cell is going to engulf the nanoparticle delivery system with a portion of their cell membrane. So they form an endosome, and then they're going to escape by lysosome escape. The lysosome escape works by proton sponge effect because the tumor microenvironment is slightly more acidic than normal cells. So the membrane is going to get protonated and destroyed to release what's inside, which is the DTA and OVA. And the OVA works just the same process, but in the end, it's going to bind to the MHC class 1 molecules on tumor cell surface because OVA needs to bind to an MHC structure so that the T cells can be activated. And the second mechanism, the second nanoparticle delivery system, is we deliver OVA to the lymph nodes. We use the similar nanoparticle delivery system to carry OVA into the lymph node by a lymph node draining, and the micelles are taken up by professional antigen-presenting cells such as dendritic cells and activate OVA-specific T cells. And part of the T cells will be differentiated into cytotoxic T cells and kill any cells that present the same antigen, which is OVA. So by combining DTA and OVA, we can overcome immune suppression and enhance long-term immunity. The results, I'm just going to make it brief because I think it's comparatively less important. So they test the cell viability after treatment with nanoparticle delivery system, and you can see from figure B that the treatment after our nanoparticles, which is PVHR is our nanoparticles, it shows the lowest cell viability, which means that it has the most effective treatment outcome. And then they use a living organism by measuring the fluorescein intensity, the mean fluorescein intensity against CD40, CD80. So CD40 and CD80 are cell surface proteins, where CD40 is a receptor and 80 is a ligand. And the mean fluorescent intensity is a way of quantification of these two proteins because it measures how brightly a cell population are stained with a fluorescent antibody specific to them. So after treatment with a nanoparticle delivery system, we can observe that the CD40 and CD80s level are the highest, which indicates a greater treatment response. And as